last 10 years, I think that 450 or 500 clinical trials failed in glioblastoma. There's nothing that works. So if you have something with this kind of results, I mean, you have to try it.
If you were to classify unmet medical needs in our medical world, it's way up there at the top. It is clearly, when you talk about less than 5% of people living 5 years, and you start comparing it to other tumors like breast cancer and other tumors, you see just how bad is our understanding of the disease and how much we really need new and more effective therapies for the disease.
I think 2 or 3 years ago, there's this paper that was published from a group at MGH where they did single-cell RNA sequencing, and they showed that chitinase 3 like 1 is, they actually wrote it like this, is the marker of the most aggressive subtype of cells within the tumor. That's how you identify the cells. They express chitinase 3 like 1. That immediately made me think that it cannot be a marker. This protein has a role. And this is the idea came that, okay, maybe this is because it's a secreted protein, maybe this is the protein that actually drives the cells to acquire this more aggressive phenotype.
Yeah, Nikos approached this from the biology of glioblastoma. I approached it from the biology of chitinases and chitinase-like proteins, and they just happened to miraculously intersect.
We managed to find that, yes, if you put chitinase 3 like 1 in the media of the cell, so in their environment, right? So then a certain subset of cells transitions from a more subtle phenotype to a way more aggressive phenotype. And they express all kinds of genes that define this aggressive phenotype. And then we did more experiments to see transcriptionally what regulates these genes. So we found a transcription factor that is directly activated by chitinase 3 like 1, and this mediates the expression of most of these genes. So there's a whole pathway that chitinase 3 like 1 uses and makes these cells become more aggressive. And aggressive means more invasive, more proliferative, and what we call more stem-like, because they keep dividing and they're forming more clones of themselves, which is very bad, obviously, for a tumor, right? I think that the very cool thing that we found is that when we use the chitinase 3 like 1 blocking antibody, and we inhibit the growth of the tumor in mice, in vivo, when we took the tumors out and checked which genes are inhibited by the antibody, we were hoping that they will be the same genes that chitinase 3 like 1 induces in the cells, and exactly this is what we found. So I think this whole circle closes, saying, yes, this is what chitinase 3 like 1 does in glioblastoma. So it makes a lot of sense to target it therapeutically, right?
It was really extremely exciting. You know, we've been studying chitinase 3 like 1 and its role in cancer for a while, but we had no idea that there was this effect on stem cells, and that chitinase 3 like 1 was taking a more benign version of the stem cell and jazzing it up to being this really bad actor in these people. And when we saw the results, the whole story fit together. It was exactly what you would have expected at multiple different levels. And so you've now got a therapeutic target, which we know already that it works with human stem cells in murine models. And you also know that it changes the differentiation of human stem cells, and you know that it has all these other effects on cancer. And you say to yourself, this has got to be the place to go to try to get a therapy that works for these people.